Hello and welcome. Let's continue with cardiac physiology. Let's go through the heart and continue with the heart. So here now we're talking heart physiology. So here now when we go through heart physiology, let's go through and let's talk a little bit about the various electrical events that we're going to see here. Now I would like you guys to know when we talk about the ability of cardiac muscle to depolarize, when we talk about the ability of cardiac muscle to depolarize, this you'll see is intrinsic. It's intrinsic. Now, this means it is the property. It's the property of heart muscle. And this ability to depolarize through intrinsic events shows us that the heart does not depend on the nervous system. So you can say the heart depolarizes and it contracts without nervous system stimulation. This ability of cardiac muscle to depolarize is, as we refer to as intrinsic. I don't know if you recall from anatomy, but this is taking us to then, very good, the intrinsic cardiac conduction system. Okay, that's what we're going to go through and we're going to talk about eventually. So here, now when we go through, so I want you to know if all nerve connections to the heart are severed, okay, if all nerve connections to the heart are severed, the heart continues to beat rhythmically. So if I go and cut your heart out and pull it out and put it on the table, your heart's going to continue to beat rhythmically, right? As we see in transplanted hearts. Nevertheless, the heart is supplied with autonomic nervous fibers. It is supplied with autonomic nerve fibers. And now these autonomic nerve fibers, what they do, they don't set the basic rhythm because that is intrinsic. What they do, the autonomic nerve fibers, they alter this basic rhythm. They alter the basic rhythm of heart activity that is set by your intrinsic factors. So the rhythm can be altered by this autonomic nervous system. And you all know that, right? Because then we get into sympathetic and parasympathetic, where we have the ability to increase heart rate. If we're talking sympathetic stimulation to the heart, if we're talking parasympathetic stimulation, you've seen then we decrease heart rate. So this autonomic nervous system can alter, right? That's all it could do is just alter. I joke around and I tell you guys, Gelota has control as well, right? 2 magic words. Most of you guys know, pop quiz. Okay, here it doesn't matter. In the class, it's a little bit different, right? So it's just, I mean, again, what am I doing? We're just activating autonomic nervous system is what's happening there, sympathetic nervous system. Okay, more so in, I guess, if you're not prepared, right? If you're prepared, you're like, oh, come on, bring it. I got this. Let's go. Okay, so let's go through and let's talk about setting then the basic rhythm. Then we'll get into that. So let's talk setting basic rhythm there. Now we talk about setting the basic rhythm. Now the intrinsic cardiac conduction system is what we're going to get into. We're going to talk about. So the intrinsic cardiac conduction system, I want you to know is what we're going to go through and we're going to talk about now. This system, you'll see the independent and coordinated activity of the heart is due to a couple of different things. So this independent and coordinated activity of the heart is due to a couple of different factors. Number one, as you can see here on the slide, it's the presence of gap junctions, right? We've gone through, we've talked about that. What do gap junctions do? Allow communication from one cell to the next cell. So you can see we said that's what's going to allow this heart to beat as a functional syncytium, as we described. So the presence of gap junctions, and then next you have the activity of the heart's in-house conduction system. The activity of the heart's in-house conduction system, which again is called the intrinsic cardiac conduction system. Which again is going to be called the intrinsic cardiac conduction system. The ICCS, you can kind of abbreviate it as there as well. Intrinsic cardiac conduction system. Now this intrinsic cardiac conduction system, this ICCS, It consists of non-contractile cardiac cells that are going to be specialized. They're specialized to initiate and distribute impulses throughout the heart. So this intrinsic conduction system consists of non-contractile cardiac cells that are specialized to initiate and distribute impulses throughout the heart. So it depolarizes and contracts in an orderly, sequential manner. So here you could see these cells not only initiate their own impulses, but they're also going to be distributing impulses. And that's going to lead to a coordinated depolarization and contraction of the heart. Now, you need to have this coordinated depolarization. You can't have one part beating before the other beating before the other. More specifically speaking, let's say you want the atria beating before the ventricles. You don't want the ventricles beating before the atria, or the ventricles starting to beat even before the atria, do their thing. So that's all going to lead to then a mess inside that heart. We're going to go through, we're going to check out. So we want that coordinated depolarization and coordinated contraction of the heart muscle. So here then when we go through, let's talk then and let's look at action potentials that are going to be initiated by our autorhythmic cells. Let's look at these action potentials that are going to be initiated by our autorhythmic cells. So when we talk autorhythmic cells, we've seen them, we've talked about them. I told you they make up about 1% of the heart's cardiac myocyte quantity. And these cells, now when we go through the autorhythmic cells, unstimulated contractile cells of the heart, neurons, and skeletal muscle fibers, they maintain a stable resting membrane potential. Unlike those cells I've just mentioned, unlike those unstimulated contractile cells, unlike those neurons I just mentioned, unlike those skeletal muscle fibers I mentioned, the cardiac pacemaker cells, or your autorhythmic cells, your neural, we refer to them as another name there as well, neurocontractile. I got brain infarct now, can't remember exactly what that was, but you can go back and review that there as well. Now, these cells, these autorhythmic cells, so unlike all those other cells that I've just mentioned, these cells, they make up, number one, this intrinsic conduction system. They have unstable resting potentials. They have unstable resting potentials. These unstable resting potentials continuously depolarize. They continuously depolarize. They're always going to be drifting slowly towards threshold. And you all know what happens when you hit threshold, leading to an action potential. So they have unstable resting membrane potentials. We're going to give that unstable resting membrane potential a name. To get more specific, we're going to call it a pacemaker potential. I think you've picked up kind of you've seen these different terms being used before as well, right? Or we can refer to them as pre-potentials. Pre-potentials. So now with them having these unstable resting membrane potentials, this causes them to get to threshold. These spontaneously changing membrane potentials are called pacemaker potentials or pre-potentials. They initiate the action potentials that spread throughout the heart. They initiate the action potentials that spread throughout the heart to trigger its rhythmic contractions. These action potentials we're going to see are going to have three parts. So there are three parts to an action potential in relation to our autorhythmic cells or our pacemaker cells. So here we can go through and we can see those three parts. We've got a pacemaker potential, which leads to then depolarization, then leads to repolarization. So the action potentials here are made up of three parts. Let's go through and let's check out those three parts. So here, number one, you can see we have the pacemaker potentials. So here we could see that voltage, the resting voltage, not even really resting, but this voltage here is going to be about minus 60 millivolts, the resting voltage. So here, as soon as the voltage reaches minus 60 millivolts, you can see here what happens then is the pacemaker potential is going to kick in. Now, when we talk about the pacemaker potential, they're going to occur because of special properties of the ion channels in the sarcolemma. These pacemaker potentials, they occur because of special properties of the ion channels in the sarcolemma. Now, in these cells, hyperpolarization at the end of an action potential, both it's going to close. So it's going to do a couple of different events here. So again, in these cells, hyperpolarization at the end of an action potential both closes potassium channels and it opens up slow sodium channels. You know the key part there. This influx of sodium now, right? That's the key part. This influx of sodium is going to cause the inside of the cell to become less and less negative. Or you can say again, more positive, taking us to threshold. Next event, when we hit threshold, you know, depolarization is going to occur. So when threshold is reached, which is about minus 40 millivolts, calcium channels now open. Calcium channels open, allowing for massive amounts of calcium to enter from the extracellular space. As a result, in autorhythmic cells, it is the influx of calcium. It is the influx of calcium rather than sodium, which you have seen many times before, that produces now the rising phase. That produces the rising phase of the action potential and reverses the membrane's potential. So again, it's the influx of calcium here that's going to produce this rising phase of the action potential. Third, then we have repolarization. Now, when we talk repolarization is the falling phase. It's the falling phase of the action potential. And repolarization reflects the opening of potassium channels. It reflects the opening of potassium channels and potassium efflux from the cell. Very good. Once repolarization is complete, potassium channels are going to close. So potassium efflux, right? Potassium is going to make its way out of the cell, so that's what efflux. Potassium efflux declines. And the slow depolarization to threshold begins again. Again, because of the opening of slow sodium channels. Starting basically these whole events over again, starting from number 1, going back to depolarization and then repolarization. So another type of action potential now you could add to your bag of tricks, right? Your bag of knowledge, basically there. This one is going to be really important because it's got to do with the heart, especially if you're working ER, if you're working cardiology. So let's go through and let's look at then the sequence of excitation. So there you got everything all at once, just like you see here. Let's look at then the sequence of excitation. Now, when we talk about the sequence of excitation, autorhythmic or cardiac pacemaker cells are going to be found in the sinoatrial node, the SA node. Let's go right to this image here. So these autorhythmic cells, these cardiac pacemaker cells, they're found in the SA node. They're found in the AV node. They're found in this internodal pathway. They're found making up the atrioventricular bundle. They're found making up the right and left bundle branches. They're found within the Purkinje fibers. So now when impulses are going to pass across the heart in order. They're going to pass across the heart in order, beginning with the SA node, right? The sinoatrial node. The sinoatrial node is located in the dorsum of the right atrial wall, high up in that right atrial wall, right at the opening of the superior vena cava, right at the opening of the superior vena cava into the right atrium. So just inferior to the entrance of the superior vena cava. This minute cell mass has a mammoth job. It generates impulses about 75 times a minute. So it has a mammoth job. Very important job. It generates impulses about 75 times a minute. If there was no hormonal and neural factors from our body contributing to its job basically there, that SA node would beat about 100 times a minute. So without any hormonal or neural intervention, by itself it would beat about 100 times a minute. Okay, you can see that right inside of there. Now when it beats, it generates impulses about 75 times a minute. When it generates these 75 beats a minute, we refer to this as sinus rhythm. Sinus rhythm. Now when we go through and we talk, the SA node is known as the pacemaker. It's known as the pacemaker. It sets the pace of the heart. It sets the pace of the heart because it has the fastest depolarization rate in the conduction system. Again, it sets the heart's characteristic rhythm. It sets the heart's characteristic rhythm that's known as the sinus rhythm or heart rate. Or heart rate. Now, from this SA node, these impulses are going to pass, and they're going to pass down the internodal pathway, making their way over to the AV node. And also at the same time, these impulses are going to be passing from the right side over to the left side via Bachmann's cells. Via Bachmann's cells. That's how we're going to allow now both of these atria to contract. So we have the SA node here, and we've got Bachmann's cells, basically helping to take that impulse over to the right atrium. Now, when this impulse leaves that SA node, it's going to pass down this internodal pathway and down the Bachmann cells, allowing the right atrium basically to contract and the left atria to contract. Now, when this impulse travels down, it's going to arrive here, you can see, at the AV node, right? Apple Valley node? No. Atrioventricular node. Look at the location. So when we talk atrioventricular node, so from the SA node, the depolarization wave is going to spread via gap junctions throughout the atria and via the internodal pathway to the atrioventricular node. And then again, Bachmann's bundle, Bachmann's bundle or Bachmann's cells will allow this signal to make its way over. So here, basically, if we were to talk technically speaking, this is what we're going to have. I'll just draw it in for you guys, kind of give you a good illustration. You can see the pathway here they've drawn in with these arrows. It's basically rows of cells, kind of these autorhythmic cells kind of running this way. And then eventually what happens is they're all going to be communicating with these contractile cells. So they're going to allow then those atria to contract. So here you're going to have another, you can see this, basically they're depicting all of this conduction system in yellow. So I'm going to use this red kind of marker here and take you over to this side here. Now here then we're going to see little branches kind of coming through here, and these branches are going to allow the atria to contract. So this is Bachmann's bundle, is what you're seeing there. Okay, here they're showing us that internodal pathway, allowing the signal to make its way down to then the AV node. Now here as the signal makes its way down, so from the SA node, the depolarization wave is going to spread via gap junctions throughout the atria, and then via the internodal pathway to the AV bundle. I'm sorry, to the AV atrioventricular node. So via the internodal pathway to the AV node. So here we are now at the AV node. Now here at the AV node, the impulse is delayed, you can see here. The impulse pauses. It's delayed for about 0.1 seconds. It's delayed for about 0.1 seconds. And what this does is it allows the atria to respond and complete their contraction. This pause is going to allow the atria to respond to the electrical event there and complete their contraction before the ventricles contract. Now from the AV node, the signaling impulse, it passes to the atrioventricular bundle. It passes through the atrioventricular bundle, or the bundle of His, it's also known as. This is the only connection. This is the only connection you have between the atria and the ventricles. So this bundle of His is the only connection between the atria and the ventricles. No gap junctions exist there. So it transmits the signal to, then you can see here, the right and the left bundle branches. To the right and the left bundle branches. Signal, the impulse passes through the right and the left bundle branches, coursing along this interventricular septum, right, towards the apex, where then they go towards each ventricle's walls as the Purkinje fibers. So these Purkinje fibers, you see here, also known as the subendocardial conducting network. And they complete this pathway. They complete the pathway. So I want you to think here for a sec. If we're to go through and look at both of these bundle branches, let's say for example, which bundle branch would have more of a serious consequence if it was blocked, if it didn't allow the signal to pass through like it normally should? The right side or the left side? Now, before you think about the answer, I want you to think of we discussed the heart as being two pumps that are found side by side, I keep telling you guys. Now, tell me, is the right pump, I mean, they're both important, obviously, but which one has a more important task there? It's going to be the left side, because the left side has to pump blood through the body. It has to make its way, the blood has to be able to make its way all the way to the top of the skull, all the way down to our toes. Here, we saw blood is basically going to get routed into the pulmonary trunk and make its way then from that pulmonary trunk into the right and left pulmonary arteries and then eventually right into the lungs. So there, gravity is, you can see the only kind of component there that's going to be allowing this blood to kind of trickle forward when it makes its way to a certain point in those arteries. Okay, so here we can have that pump take place, okay, or the blood can make its way rushed into here. I mean, obviously that can eventually become detrimental as well. It depends on the degree of blockage there as well. If you have a total blockage, obviously then this patient's going to require invasive care. But now if it's a mild blockage, now there usually you'll see then it's not going to be as detrimental. And here then the left side has that more important task there. I have what you call a right bundle branch block. This right side is not conducting impulses like it should be conducting impulses. So a lot of things I shouldn't be doing, but like you guys tell me, YOLO, right? You only live once. So yeah, but here now let's move through and let's talk then modifying this basic rhythm, modifying the basic rhythm. So here before we take off, you can see we went through basically these different parts here. Okay. So get that checked off there. Let's talk about modifying then the basic rhythm. Now when we talk about modifying the basic rhythm, here is what we're going to get into. But before we jump into that, let me show you now, since we've gone through different parts of the heart, we have seen now, okay, different action potentials. So here you can see the pacemaker potential in the SA node when you look at just the SA node. Here you can see what's happening in the atrial muscle then. And then here, AV node, pacemaker potential we've got here. And then here you can see ventricular muscle. And we've talked about basically what we've seen here. Right, this famous plateau. So here then we could see all the different action potentials basically in relation to the different parts of the body. And now here when we go through and we check out the next part here we're going to get into talking about modifying basic rhythm and checking out now extrinsic innervation of the heart. Now when we go through and we talk about extrinsic innervation of the heart, although the intrinsic conduction system is going to set the basic heart rate, we said, fibers of this autonomic nervous system modify, as we described before as well, they modify the beat. Not only do they modify the beat, they also introduce a variability to heartbeats. The sympathetic nervous system, also known as the accelerator, you can think of. We can come right over here. This picture describes it really well. So the sympathetic nervous system, or the accelerator, you could think of, it's going to be responsible now. So here you can see it's going to be responsible for increasing heart rate, as we've described before. So it increases both the rate and the force. These are two different things. The force of the heartbeat. Is this heartbeat a strong heartbeat, or is this a very faint heartbeat? Okay, so it's going to modify both the rate and the force of the heartbeat. When we talk parasympathetic nervous system, the parasympathetic nervous system, when it gets activated, you can think of as the brakes. It slows the heart. So let's look at now the cardioacceleratory center. Now the cardioacceleratory center is going to be found located in the medulla oblongata. And this center, it carries the sympathetic component. This system, it innervates, as we can see here in the picture, it innervates. You can see the SA node. It innervates the AV node. It innervates the muscle, the heart muscle, as we can see there. And it innervates the coronaries. And it innervates the coronaries. It causes the coronaries to dilate. And when they dilate, what's going to happen? It's going to bring in more blood to the tissue. Now, when we talk cardioinhibitory center, the cardioinhibitory center is going to be found also at the level of the medulla oblongata, and it conveys inhibitory impulses. It conveys inhibitory impulses for this parasympathetic nervous system. Now, this system, you can see here, this parasympathetic system, it's going to be coming in and it innervates the SA node, as we can see there, and the AV node. So what does it do, we said? It's going to decrease. It will decrease heart rate. So here we can see the actual fibers coming from the dorsal motor nucleus, and it's the vagus nerve we can see there that's involved with that parasympathetic innervation. Next in here, we can move and we can check out then what we call electrocardiography. Here we're going to check out basically an EKG or an ECG, it's also known as, or an electrocardiogram. First, let's talk electrocardiograph. An electrocardiograph, because we're talking electrocardiography, so an electrocardiograph, okay, without the I, an electrocardiograph, it's a device. It's a device that detects electrical currents generated in and transmitted through the heart and spread throughout the body. So again, it's a device that detects electrical currents generated in and transmitted through the heart and spread throughout the body. So an electrocardiogram or an EKG or an ECG, that itself then is a graphic record. That's an actual graphic record versus the machine electrocardiograph. So here we have an electrocardiogram now. This electrocardiogram, this EKG, is a graphic record of heart activity. It's a graphic record of heart activity. We'll see an ECG is a composite. It's a composite of all action potentials. It's a composite of all action potentials generated by nodal and contractile cells at a given time. You'll see, to understand an EKG, or I'm sorry, to actually do an EKG, to record an EKG, we'll talk first, there's going to be electrodes. There are 10 electrodes that are used for what we call a 12-lead EKG. And these electrodes are going to be placed at various sites of the body. If you've ever done some, you've seen 6 will go around this area here, and then 2 will be in relation to the arms, and then 2 in relation to the legs, basically, we have there as well. And now I'm sure there's more advanced ways of doing that stuff. So here when we go through, we talk EKGs. Now, a typical EKG is going to have, now, so let's talk what an EKG is going to look like. An EKG is going to have, you'll see 3 almost immediately distinguishable waves. So let's actually go to the picture. So here's one recording where it depicts to us a few characteristic or distinguishable waves. The first wave you can identify or distinguish here right off the bat is the P wave. The P wave. Next here we can see the QRS complex. This QRS complex. And then third, you can appreciate that T wave there. So 3 things you can see right off the bat. Let's talk first P wave. P wave. When we talk P wave I'd like you to know is the first wave. As we can see here, it's the first wave we're going to see in an EKG. And this P wave, it results from the movement of the depolarization wave from the SA node through the atria. So the first wave, this P wave, it results from the movement of the depolarization wave from the SA node through the atria. You can see then about 0.1 seconds after the P wave, so about 0.1 seconds after the electrical event, after the P wave, the atria will actually contract. Then we can see here we have the QRS complex. Now when we talk QRS complex, the QRS complex is going to result from ventricular depolarization. So we had atrial depolarization for the P wave. Ventricular depolarization is creating this QRS complex. The QRS complex again results from ventricular depolarization. It has a complicated shape because the paths of the depolarization wave through the ventricles are going to change continuously. So it creates this characteristic QRS complex that we see here. And then when we talk T wave, you can appreciate the T wave here as well after the QRS complex. Here we can see the T wave. It looks similar to the P wave, okay, with a little bit of variations there. And when we talk T wave is going to be caused by ventricular repolarization. Ventricular repolarization. So you may be wondering, he talked about atrial depolarization, ventricular repolarization. Well, he didn't mention anything about atrial repolarization. Atrial repolarization. When does that happen? Why isn't there a wave I see here? It happens during this ventricular depolarization phase. Why don't we see it? Because this event is much more of a drastic event compared to atrial repolarization. So here we can see signal being transmitted, this depolarization wave traveling from that SA node, and you can see this P wave is already there. As the P wave goes through, you can see now the atria will contract, taking us then to this electrical event which occurs, and then the ventricles will contract, leading us then to repolarization of the ventricles. And we being at the end of the T wave. So you can see again that electrical event's going to happen split second first, and then all of a sudden the contraction is going. It's kind of like you can think of a domino effect. The dominoes are going, and then the electrical event is going, and then the contraction is actually coming right behind it. Here we can appreciate a few EKGs. The first one we can see here is normal sinus rhythm, where you can appreciate, now we'll grab this, we can't see a P wave here, but it would be like here, QRS complex, and then T wave, here's a P wave, QRS complex, T wave, 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 then P wave, QRS complex, T wave. It continues all the way through. Here what we see is we see junctional rhythm. Here what happens is the SA node is non-functional. So P waves are absent. So you can see here, there's no P wave. QRS, T. QRS, T. QRS, T. Now when we talk atria, if the atria, now why aren't they the pacemaker cells? The reason why the atria aren't pacemaker cells is because when they beat without any innervation, okay, you'll see they beat about 40 to 60 times. So with them beating only 40 to 60 times, the SA node is beating 100 times. The SA node says, wait, I'm the SA node. I'm going to be the pacemaker here. But now if it goes out, AV node is going to pick up. So it'll be basically the boss there then. Now, here we can appreciate what we call second degree heart block. Now here, you can see some P waves are not conducted through the AV node. So hence, we're going to have more P than QRS waves are going to be seen. So here now, again, some P waves are not going to be conducted through the AV node. So here is what we call second degree heart block. So let's grab this one here first. So P wave, QRS, T, P, P, QRS, T, P, P, QRS, T. Okay, so you can see that there as well. Now here is ventricular fibrillation. They need intervention. They need intervention stat. Okay, chaotic, grossly irregular EKG. You can't tell P wave, QRS, even T wave how they're supposed to be. Okay, so this can be seen in acute heart attacks and also in electrical shock patients as well. Now we also have PR interval, ST segment, and the QT interval. So you can go through and you can check those out and understand what we have going on there here as well. So longer events, usually, or events taking place in between other events is kind of what you see there. Let's talk then heart sounds. Heart sounds. Now we talk about heart sounds. I want you guys to know heart sounds are going to be associated with the heart valve closing. With the heart valves closing. And these heart sounds are going to be described as lub, dub, pause. Lub, dub, pause. And so on. With the pause now, the pause indicating the period when the heart is relaxing. Indicates when the heart is relaxing. Now the first sound is going to occur as the atrioventricular valves close. As the AV valves close, we hear the lub sound. The dub sound is going to occur now. That's the second sound. It occurs as now the semilunar valves are going to close. As the semilunar valves snap shut. Now when we talk murmurs are going to be abnormal heart sounds. They're usually going to be indicating an incompetent or a stenotic valve. And this murmur is going to create turbulence in our blood flow. So here you can actually see where we can auscultate the various heart sounds. So for lab, what you could be doing is grab, if you have a stethoscope at home, most of you probably do or don't, you guys are in the medical field, this is something you'll probably start buying and investing in. And here we would actually be using them in class and doing the lab where you'll grab a sphygmo and check a patient's or your partner's blood pressure, and they'll do the same to you. And you will actually have to do the labs there as well. And you can do the same thing. Grab our labs from our website, and you can do basically the same exact thing there that we would set up. There's nothing here in this lab that requires something really different from what you could find. Again, the stethoscope and the sphygmomanometer are going to be the two things there, but after that the events are going to be basically you checking pulse, checking respiration, and getting blood pressure down there as well, either sitting, standing, laying down, and then check the same things before you do exercise, and then after a couple of minutes of exercise, and so forth. And then we also have a few other labs in there. You can kind of go through and take care of those as well from the website, cardiovascular labs. Now here we could go through and we can check out now the different areas where you would actually place your stethoscope in order to be able to appreciate these valves and to hear them by themselves. Because if you go and you put your stethoscope right in the middle of the heart, you're going to hear all four valves, and you're going to hear the whole machinery basically working together. So here what we do is we either go upstream or we go downstream from the valve, and you can isolate that valve there and hear it better. So if you want to listen to the aortic valve you can see here is going to be best heard at the second intercostal space, right parasternal, basically. Right parasternal, right Right side first, right parasternal. Para, right prefix next to, and the sternum. So that's what you see right inside of there. And here you can see that valve. The aortic valve is going to be again on the left side. So to hear it best, you'd place your stethoscope right inside of there. Opposite side now, left parasternal, second intercostal space, pulmonary valve. Pulmonary valve. And then down here we can appreciate tricuspid. And then we can appreciate mitral valves as well. So tricuspid, you can listen to. It's telling us sounds are typically heard in the right sternal margin of the fifth intercostal space. I've also listened to them here in the left sternal margin of the fifth intercostal space as well. You can listen to that valve right there. And now mitral valve, so tricuspid here or left side or right side. But same exact location, fifth intercostal space. You can see parasternal. Mitral then, fifth intercostal space. Now in line with middle of the clavicle. So when we're talking middle of the clavicle, what are we talking about? Very good. Awesome. I knew you knew that. Mid-clavicular. Okay? So you got to remember those landmarks from anatomy. So mid-clavicular. So that's where you'll place your stethoscope and listen to these valves individually. Let's check out then the mechanical events, and we'll look at the cardiac cycle. And we will look at the cardiac cycle. So here now, when we go through and we talk about the cardiac cycle, I would like you to know here, we have now, all right, so let's look at, again, the cardiac cycle now. All right, so when we go through and we talk cardiac cycle, I want you guys to know that the heart undergoes dramatic movements. It undergoes dramatic movements. It contracts, and when it contracts, it forces blood out of its chambers. And then it relaxes, allowing its chambers to refill with blood. Now, when we go through and we talk the cardiac cycle, there's two terms here that I'd like you to be familiar with, and those two terms we can appreciate here are going to include systole and diastole. Now, when we talk systole, this term systole, it basically is talking about when the heart contracts, forcing blood out of its chambers. So it's a period of contraction. Now, when we use the term systole, okay, you have to understand contraction is taking place and blood is being basically forced out of its chambers. Now, the next term is diastole. Diastole is the process when the heart relaxes. It's the process when the heart relaxes, and when the heart relaxes, this allows its chambers to refill with blood. So diastole is the period of relaxation. Now, when we go through and we talk about blood basically flowing through the heart, you'll see the cardiac cycle is going to be talking about how blood flows through the heart. And what this cardiac cycle is it's a series of pressure and volume changes. We refer to them in, you guys get deeper into your medicine, you'll see PV loops, pressure volume loops. When you get deeper into medicine. Madison, I'm saying medicine. Medicine. Oh, I guess my pronunciation is horrible. Okay, so now when we talk cardiac cycle, the cardiac cycle is going to show us all the events that are associated with blood flowing through the heart during one complete heartbeat. During one complete heartbeat. Now the cardiac cycle is a succession or a series of pressure and blood volume changes in the heart. The cardiac cycle is a succession or a series of pressure and blood volume changes in the heart. So here what we're going to do is we're going to move over right to this image here. Now in this image here we can see basically pressure right inside of this chart, right inside of here, and then here we can see volume. Okay, when you guys move through and this is like the preschool way of explaining this. You guys will see there's just going to be basically something that's going to look kind of like this. And it's going to be found in a graphic similar to something like this. You have pressure on one side, volume on another side, and it's going to be showing you exactly what's going on. So like I said, this is a more simplified version of that. So here now when we go through and we talk about these pressure volume changes, you can appreciate those changes now that are going to be taking place here within these graphics. Now here on top we have a couple of different components being depicted here. Here we can see we have first the electrocardiographic readings. Okay, so then we can see your heart sounds. So this is all going to be correlated with each other. Okay, then coming down to you actually seeing what's happening in relation to the valves, AV valves, semilunar valves, and then this is what we're going to get into. We're going to define and describe basically all these events that are taking place in this phase, all in here. Okay, that's what you should be able to talk about. Same thing then in this like brownish khaki color area right in here, and then same thing with the brownish area here, and then all the way down here in the blue again. So here let's go through and let's talk first ventricular filling. That's what we're seeing here. Okay, so ventricular filling. Now when we talk ventricular filling, I'd like you to know when we talk ventricular filling is going to be taking place mid to late diastole. This is the during ventricular filling, we're going to be looking at the part of ventricular filling where we are looking at the phase from mid to late diastole. Now here I want you to know that the pressure in the heart is low. So pressure in the heart is low, and you can see that right inside of here. Okay, pressure pressure levels you can see in the different segments of the heart. So you got to make sure you know what you're looking at. They're showing us the aorta here. They're showing us the atria here. It's the left atria, and then here we can see the left ventricle. So you can see all three of those important chambers we've got right inside of there as well. So here we can see now when we talk ventricular filling, when we're talking from mid to late diastole, pressure in the heart is low. Now blood returning from the circulation is flowing passively. So we're going to see here the volume is going to be increasing. So blood returning from the circulation is flowing passively through the atria and the open AV valves into the ventricles. And the aortic and pulmonary valves are closed. We'll see about 80%, we're going to see about 80% of filling occurs during this period. About 80% of filling is going to occur during this period. The remaining 20% is going to be delivered to the ventricles when the atria contract. And that's going to be towards the end of this phase. That will be towards the end of this phase. So now the stage is set for atrial systole. Okay, following depolarization, the P wave on the ECG, or the EKG, the atria contract. When they contract, you can see that event right inside of here now. The atria contract. And this compresses the blood in their chambers into the ventricles. This will compress the blood in their chambers into the ventricles. Now at this point, the ventricles are in their last part of diastole. At this point now, the ventricles are in the last part of diastole, and the maximum amount of blood, okay, the maximum volume of blood you can say that they will contain in the cycle is in there now, and that volume of blood is going to be given a name. It's going to be called the end diastolic volume, or the, you can see here, EDV, the end diastolic volume. Okay, then the atria relax. And the ventricles will depolarize. So let's look at all that again. That should be characterized by the P wave. Bam. We've got that marked. Okay, so here what happened, the atria, you can see their pressure is decreasing, and all of a sudden the pressure goes up. Why did that pressure go up? The pressure went up because they contracted. And then what happened? You can see here, blood was filling, and then it came kind of to a point here, and then oomph, it filled up even more. You got that push. And you can see that being indicated right inside of here. So we said then the atria will relax and the ventricles will depolarize. So we're going to look at the QRS complex in relation to the ventricles depolarizing. The atria are going to be in diastole. So atrial diastole will persist through the rest of the cycle. So let's look at then ventricular systole. But before we jump down into ventricular systole, I'd like you to see now, this is where we're going into 2A now. Okay, so we're going into 2A. We just finished up to this blue segment here, basically. Now, when we talk 2A, so you can see here that the electrical event is going to be basically depicted here, that QRS complex ventricular relaxation, ventricular contraction. So let's look at ventricular systole. As the atria relax, okay, as the atria enter diastole, one way I remembered it was diastole, die, you're dead, you're not moving. So diastole was one way, I remembered, okay, this is where there's a movement happening. And then the other is the other. That's the easiest way to remember things. So when we go through and we talk ventricular systole, now the ventricles begin contracting. Rising ventricular pressure is going to close the AV valves. So here you can see now when the blood fills into the ventricles, this will cause the pressure in the ventricles to increase. This rising ventricular pressure is going to cause the AV valves, basically behind them you can think of, it'll cause those valves to close. Now this phase ventricular systole, we can divide this phase up into two parts. First we have here what we call the isovolumetric contraction phase. The isovolumetric contraction phase. Second, then we'll look at the ventricular ejection phase. So let's look at the isovolumetric contraction phase first. Now, the split second period when the ventricles are completely closed chambers and all the blood in the chambers is going to remain constant, this is going to lead to basically the pressure increasing. So when we talk isovolumetric contraction phase, okay, this is the phase, this is the split second period. It's not lasting for too long. It's a split second period, and it's a period when the ventricles are completely closed chambers. So we said the rising pressure causes the AV valves to shut. The semilunar valves haven't shut yet, because remember we said that the pressure, when it gets higher on this side compared to the other side, that's going to cause it to open or it's going to cause it to close. So here we've got enough pressure to cause the valve back there to close. Remember, because of the anatomy there as well, that helps out. Now we're going to see all of the chambers are going to be basically these ventricles, these chambers are going to be completely closed. Okay, so blood is already filled in there, and they're completely closed, and now pressure starts to rise. So the pressure starts to rise. Now this is going to take us to the second part then, the ventricular ejection phase. Now when we talk ventricular ejection phase, now this is when ventricular pressure exceeds the pressure in the large arteries. That's going to cause the isovolumetric stage to end and the semilunar valves to snap open or pop open. So the semilunar valves will be forced open and blood then is going to be expelled into the aorta and the pulmonary trunk. So first, let's go back to isovolumetric contraction. Let's break that word down first. Iso, the same, volumetric. So the volume is staying the same. It's the contraction phase. Basically, you can think of. So here what happens is the volume, let's look at that. Look at the volume was 120 milliliters of blood before this phase began. And it's 120 after as well. So that's why it's isovolumetric. The volume is not changing. Iso, the same volume. Okay, but what is changing? Let's look at this up here. Look at that pressure. Okay, we're going to talk left ventricle. That's the main structure. That's why they've mentioned that here as well. Look at the left ventricle. So now the blood is all inside of there, right? This is that isovolumetric. But we didn't say isopressure. No, pressure we said is increasing. So you can see here that pressure is increasing. That pressure needs to be higher than the pressure in the aorta, right? Isn't that what we said? That's what's going to cause then the valve to open. So look at what's going to happen right when we pass that there. Pretty cool, pretty fascinating. So look at when that pops open, it opens the valve. Look at right down here. What's going to happen? Look, come all the way down to the volume. All that blood is going to be ejected out. So volume, we're going to go from about 120 milliliters of blood all the way down. We're going to see to about 50 milliliters of blood. So the ventricular ejection phase now will allow that volume of blood out. So here now we can see heart sounds correlated with that event as well. So when you talk first sounds, AV valves, we said snapping shut. That's what we saw taking place right inside of here. And then semilunar valves, we're going to see snapping shut as well. And then again, the EKG you can follow with the electrical events there as well. So let's move now to the isovolumetric relaxation phase. Now this, we're going to look at early diastole. Now during this brief phase, again I'm mentioning that as well as I did during the isovolumetric contraction. So during the isovolumetric relaxation phase, during this brief phase following the T wave, the ventricles will relax. The ventricles relax because the blood remaining in their chambers, referred to as the end systolic volume, is no longer compressed. So ventricular pressure drops, and blood, ventricular pressure drops, and blood in the aorta and the pulmonary trunks are going to flow backwards towards the heart. So once we've contracted the ventricles, ejected that blood out, remember we said gravity is going to allow some of that blood to trickle back. That's what's going to increase the pressure then on that side compared to this side, causing the valve to snap shut. So again, we're in the isovolumetric relaxation phase. The isovolumetric. Look at the volume is not changing. We're just relaxing. So you can see that right inside of here. Look at the volume is not changing. We're going 50 to 50. But look at the pressure. The pressure is coming down. We said following that T wave. So ventricular pressure drops and the blood in the aorta and the pulmonary trunks flows backwards towards the heart, closing the semilunar valves. Now, when the aortic valve starts to close, when the aortic valve starts to close, it rises aortic pressure briefly. So when that blood is trickling back onto that aortic valve, look at here, what's going to happen is it's going to cause aortic pressure to rise briefly, creating this dicrotic notch. And then you can see that pressure just slowly starts to decrease again. And then here we're going to see volume start to fill again. We'll look at that in a few seconds. Now if the heart beats about 75 times a minute, the cardiac cycle lasts about 0.8 seconds. Atrial systole we describe as lasting for only 0.1 second. Ventricular systole lasting for about 0.3 seconds. Then the remaining 0.4 seconds, is going to be a period of total heart relaxation or diastole. We call that the quiescent period. Also, again, I want you to understand 2 points here. I want you guys to understand 2 points. Number one, that blood flow through the heart is controlled by pressure changes. Number 2, blood flows down a pressure gradient. Right? It goes from high to low. The right and the left hearts, they're essentially the same, except for pressure. Except for pressure and the location of obviously where their blood is coming from and where it's going. Okay, but the big difference there is going to be pressure. The pulmonary circulation, our pulmonary circulation has a pressure of about 24/10 millimeters of mercury of pressure, while our aorta has about 120/80 millimeters of mercury of pressure. So when we check blood pressure, we check blood pressure in an artery that's closest to the heart, and when we check blood pressure in the artery that's closest to the heart, it is about, we say average should be about 120/80 millimeters of mercury. So this is where we'll stop.